I will like at least briefly touch on all these things. So yeah, what is a cell and cell theory? So there are two cell theories actually. One of them is the traditional cell theory and one of them is the modern cell theory. They're not actually like, they're not separate. Like you don't consider the modern one and get rid of the traditional one. They're both kind of, you have to consider them together. Modern one kind of just adds on to the traditional cell theory. So basically all life is composed of one or more cells. A cell is a basic unit of life and all cells come from other cells. But then like to get a little bit more complicated, like energy to sustain life is generated within the cell. So the cell doesn't obtain energy from outside, it kind of makes its own. All cells have to contain DNA and cells in closely related species function similarly to each other. Yes. Yeah. So what's in a cell? This is an important distinction to make. Is mostly between the cytoplasm and the cytosol. So the cytoplasm is everything that's in the cell. So that includes the cytosol and the organelles. The cytosol is just the kind of gooey gel-like stuff that's within the cell that organelles are suspended in, whereas the cytoplasm is basically everything that's in the cell. And organelles are things like mitochondria and stuff like that that have a function within the cell. And they can be membrane bound or non-membrane bound. Just a side note, inclusions, they sometimes like to mention inclusions and they'll be like, what the fuck is an inclusion? They're basically just random cell products. Like if a protein has just been made and it's like being transported around a cell, it's called an inclusion. Awesome. So this is kind of an overview of the organelles we're going to go through. The ones on the left are kind of the basic ones that you would learn in Year 12 Bio. The ones on the right are ones that you probably would have slightly covered, but probably haven't covered it in as much detail, but I will be going through. So firstly is the cell membrane. Oh, we didn't learn peroxisomes. Yeah, awesome. Peroxisomes, they kind of just like mention it every now and then. It's nice to know what it is, so I will cover it a little bit. So yeah, so the cell membrane, what you need to know is that it is a phospholipid bilayer. Something they like to mention is the fluid mosaic model, in which like basically the whole thing can kind of move and shift about itself. Like if you think about it, the two sheets of the plasma membrane can actually move on top of each other as well. The chemistry behind it is like the non-polar tails and the polar heads, which cause the polar tails to point inwards. So inside the membrane, it's hydrophobic, whereas outside it's hydrophilic. That does make it impermeable to a lot of water-soluble molecules. Yeah, and these are some contents of the plasma membrane. So you obviously have the phospholipids. Cholesterol just kind of is there to provide rigidity. Proteins are kind of things like you have protein channels and you have like protein receptors and stuff like that. Protein channels are basically like these little channels that facilitate transport of ions and stuff in and out of the cell. There are a lot of different types of protein channels, but to be honest, it's more of like a Year 12 Bio kind of concept that you learn rather than something you go into a lot of depth in med. So realistically, like after Sem 1 of Year 1, you're not going to need any of your biochemistry, which is a bit of a rip, but you have to learn it anyway, which is, once again, a bit of a rip. Carbohydrates, usually it's just like a sugar that will stick out the cell membrane and be like a receptor for like hormones or other proteins and stuff like that. The nucleus, hopefully most people know what the nucleus is. It contains DNA, basically. An important distinction to make is that when the chromosomes are just like, not when the cell isn't going through division or anything, the chromosomes are actually long strands of chromatin. And then once cell division starts, they get a lot of like signaling and they become chromosomes. Yep, all human cells are diploid and have 23 pairs of chromosomes. Yeah, so the nucleolus. You don't really need to know that much about the nucleolus. The main thing is that it has a lot of ribosomal RNA, which is basically like, it's similar to DNA, but for ribosomes. And basically creates ribosomes. And ribosomes are pretty important. We will talk about those in a second. Yeah, so just remembering the differences between the nucleus and the nucleolus, just to make sure you don't get them mixed up. The nucleolus is within the nucleus, mainly. The mitochondria is the powerhouse of the cell. Hooray, we love that. Yeah, so it literally is powerhouse of the cell because it basically just, it's the site of where all the ATP in basically your whole body is made. You have cells everywhere, obviously, and each of those cells has, most of those cells should have mitochondria. And during aerobic respiration, it produces ATP. As you can see in the respiration equations at the bottom, the plus energy is ATP. A thing to note is that mitochondria have double membranes, and that's because the theory is that there used to be their own organism, like back before sentient life was kind of formed. They used to be separate, and then bigger cells basically engulfed them, and as they entered the cell, they kind of gained that extra membrane of like what the original cell was made of, kind of thing. So now they have two membranes. Yeah, so biphagocytosis. And another side note is you inherit mitochondria from your mothers, which seems really random, but you know, sometimes they like to test you on it. The Golgi Apparatus. Basically, they call it the post office of the cell. Things will come in on the cis face. So there are two faces. The cis face is like, I like to think of it as like, so the cis face and the trans face. The cis face is the bigger one. Trans face is kind of the smaller one. I like to think of it as the trans face transmits like packages of stuff to other places, and the cis face kind of receives stuff. Yeah. Ribosomes. So I was looking back at the lectures and I really wanted to push this. Ribosomes are not membrane bound, so they're not considered organelles. But you know, they're very important in the cell for protein synthesis. And something that confused me for a while is that there are two subunits of a ribosome. They don't tell you this in school, but there are two subunits. One is a smaller subunit, one's a large subunit. Their names can vary depending on if you're looking at prokaryote or eukaryote, as you can see in the picture. In prokaryotes, it's the 30 and 50s subunit, whereas in eukaryotes, it's 40s and 60s. Those two subunits have different purposes, but we will get into that kind of when it's more relevant towards protein synthesis and stuff. Yeah, see, there's kind of conflicting information about it. Yeah. So there's a question from James that says, organelles can be membrane bound or non-membrane bound. Because the thing is, it's kind of, I'll answer the first question first. I feel like it's kind of vague because like technically in a prokaryote, do they have quote unquote organelles? Like I feel like there's still, people still like calling them organelles, but you know, yeah. It can be a hard distinction to make. Like I personally would call ribosomes an organelle, but like the lecturer didn't want to call them organelles. And if you look it up on the internet, they still say ribosomes are organelles. So it's just kind of a lot of conflicting information. I just took that from the lecture slide purely because what they examine you on is basically what's on the lecture slide. So just kind of an important point to make there. But realistically, there's a lot of conflicting information and it's hard to figure out what you take as the truth, I guess. Yeah, exactly. So yeah, it's a bit fluid around the naming stuff, but I think the more important thing is to know like what that ribosomes are there and that they're important. In terms of memorizing sizes of subunits, it's not really that important. It's more like when you start doing pharmacology, they'll be like, oh, this affects the 30th subunit of, you know, the bacteria. So it's nice to kind of know the context behind it. Yeah, so more about ribosomes. It's probably not this important, quite important yet. I will be going over it again when we get to protein synthesis, but within the larger ribosome subunit, there are 3 sites, and it's basically how translation is facilitated. Yeah, so the rough ER, yeah, it has ribosomes embedded into it, has sacs called cisternae. It's called rough because it has ribosomes in it, and then the smooth ER doesn't have ribosomes. Yeah, so it's a site of protein synthesis because it has so many ribosomes. And once enough protein's kind of made there, they'll ship it off to the Golgi apparatus. And also when they're often immediately found around the nucleus, purely because if you think about it, you have DNA replication that happens in the cell, and then if you want the strand of like mRNA to leave the cell, sorry for people who haven't done bio, or like if you have done the bio bridging course, it might make some sense. But when the mRNA leaves the cell, it doesn't want to like have to travel ages to find a ribosome. It wants to kind of it to be right there, because otherwise, you know, it could get more difficult. So that's why they're right around the nucleus. If they ever tell you to draw a cell, draw the ER around the nucleus. Smooth ER, once again, similar to rough ER. They have slightly different structures. Yeah, so it's more tubular versus like flat. Yeah, main functions are synthesis of lipids, detoxification of, oh, this construction stopped, hooray. Detoxification of byproducts, and also storage and metabolism of calcium ions within the cell. I don't think that last point is very important because I don't really learn that much about calcium, but you know, fun note. The cytoskeleton. Okay, so you probably have heard of the cytoskeleton as just like, it exists, like it's there, don't mind too much about it. But they really like to go into the cytoskeleton. So if you think about it, the main function of the cytoskeleton is actually to move organelles within the cell. So I guess the way you can think about it is almost like they're rails, like suspended rails. So like a mitochondria would like attach to one, and if it wants to move around, it'll move via the cytoskeleton. Yep, and there are three major components of the cytoskeleton. So yes, these are the main components of the cytoskeleton. There are three. You have microtubules, intermediate filaments, and microfilaments / actin filaments. You probably don't need to know this in like too much detail, but they do like to bring it up as kind of one of those more niche questions. Yeah, so microtubules are the largest. It kind of is confusing between microtubules and microfilaments. I guess the way you can think about it is a tube, if you look at a tube visually, it can look quite big, whereas a filament is like a filament of like, what do they call it? Like, I just immediately think like guitar strings, but like they're pretty fine. So just think like tubes are kind of chunkier and filaments are like very fine, and then intermediates in the middle. So like, yeah. Yeah, so microtubules are hollow tubes made from tubulin dimers. Intermediate filaments are like coiled into cables, and actin filaments are like basically just strands. Actin does become quite important when you come to muscles and how muscles work and stuff. That will come later, but just keep the word actin in mind because it will come back. Centrioles. So now we're starting to get into the bit where we didn't really learn this in neutral bio in this much detail. So centrioles, they're formed from 9 microtubule triplets. So remembering structures is actually pretty important because in following slides you will see that flagella and cilia have quite unique structures that they really love to test you on. So just kind of remember the difference between this structure and the following structure. So this is from 9 microtubule triplets in like a pinwheel structure. And the centrioles are always found. So like one of these boys will be perpendicular to another one. And they kind of just float around in right angles. And these are used for spindle formation during mitosis. Flagella. So flagella are like these long wiggly things that help cells move around. Yeah, so it's like what you find on sperm cells. Like they help the cell move. The important thing is that it's this 9+2 structure. So you can see in this picture here, let me just use this mouse. It has 9 pairs on the outside and then 2 on the inside. So that's what the 9+2 structure is. It is quite important because they will test you on it. They'll be like, what has a 9+2 structure? And you have to remember it's mostly flagella, but also cilia can have a very similar structure. Yeah, so cilia, it's like what you can find it in your respiratory tract. So the cilia will basically these like finger-like things or like hair-like things that basically just move back and forth. So the difference between flagella and cilia is that flagella like really move. Like they're like It's hard to explain over video cam, but like, yeah, it's like a sperm tail, whereas cilia are just like almost like as if they were hairy bits that just line your, like they line your mucus tract and stuff like that. Like they don't actively move things, like move a whole cell, but they will do things like helping to move mucus up back into your throat so you can cough it out. There are a lot of different types of cilia. You really don't need to know that much about it. But yeah, non-motile cilia. There are motile and non-motile cilia. Microfilaments. So there are actin and myosin filaments. And once again, this does become quite important in muscle cells. Actin, there's a lot of, like, you have much more actin generally in the body than myosin. You only really find myosin in muscle cells. So yeah, and actin, yeah, you can kind of find it around the body. It's in an intertwined double helix shape. And is important for cell motility.
Hello?
But rather than, like, to be honest, it's probably not the most important thing to remember it now, but definitely keep these in your head for when we come to muscles. Because actin and myosin just is the only thing that makes up muscle cells.
No, I'll just stop.
Lysosomes and peroxisomes. So you probably don't need to know too much about
It's
this.
literally been
Look at the
such a
picture
mess.
here.
Oh, sorry. Am I talking?
The picture has disappeared. I'm very sad. Okay, anyway.
Oh my god.
Basically, the trick is you don't really need to know in detail what they do, but peroxisomes oxidize long-chain fatty acids, and lysosomes hydrolyse macromolecules. That's literally all you need to know. That's literally all you need to remember. Like, there's not really much more to it than that. I did have a table that's the difference between lysosomes and peroxisomes, but it's disappeared. So hopefully I'll make sure that's in there for when we upload slides. Question time, hooray. So some people might have, oh, where are peroxisomes found? They're kind of just like floating around the cell. They're almost like the cell inclusions that I mentioned earlier, but they're quite small and they just literally just float around in the cytosol. Yeah. So, and it's also like, say, if the cell has received a product that needs to be oxidized, then usually it'll be like phagocytosed, so it's within a membrane-bound like pouch thing. The peroxisomes will go along into the vesicle, oxidize everything, and then leave, basically. Yeah. So, which of these is not part of the cell theory? I guess put your answer in the comments if you want to. Have a think about it. I'll give you a couple of seconds. Yeah, so the answer is C. B1, B2. What? Yeah, so the answer is C, because cells are the most basic unit of life. B is one from the modern cell theory, so technically is still part of the cell theory. Yeah, I'm not gonna lie. I could barely remember anything from microfilaments, microtubules. When I first learned it, I was like, well, why is this relevant? To be honest, it's not that relevant. And intermediate filaments, you don't really need to know that much. Even microtubules and microfilaments, they're not very high yield, is what I'd say. Like, you can remember them, but like you'd be better off remembering other things if it like came down to it. But you're more than welcome to kind of like have a look into more detail about them. Yeah, don't worry about it too much. Yeah, it's not very important to know, I guess. It's more important like later on in different contexts. Question 2. What cell has a 9+2 arrangement? Give you like 15 seconds or something. Yeah, cool. So the answer is, yep, Flagella, but also Cilia. Yep, awesome. So I'm just going to briefly cover like prokaryotes and eukaryotes. Prokaryotes are unicellular, no membrane-bound organelles, and there are bacteria and archaea. Bacteria are basically like what you find in daily life. Archaea are like really old, and they basically exist in like volcanoes and like hot springs, like basically it. Yeah, in particular, like this came up in a shoot at some point, and it was really confusing at the time because we were all like, what the fuck are these things? So flagella, obviously we've kind of covered, but things like the glycocalyx, it's basically a coating, and types of glycocalyxes include capsules and slime layers. For the longest time I thought they were all separate things, but no, they're kind of umbrella term, glycocalyx. Sorry, I've been talking for a while. Yeah. So glycocalyx is a carbohydrate-enriched coating that covers mostly bacteria, and it uses it's like a protective coat. So if it's like the bacteria is kind of making its way around the body, trying not to be spotted, so it can cause havoc. The glycocalyx is like sometimes they use it to blend in with other cells, and they can use it, I don't know, just protection from like general shit that goes on. The capsule has more defined borders than the slime layer, and the slime layer just kind of it's not as effective as a capsule. There are fimbriae, and fimbriae are I would say they're kind of similar in like shape and structure. Fimbriae and pili are very similar. They're kind of just like projections. And the fimbriae are like attachment points, so they help one cell attach from another cell. Pili, they'll call them sex pili, which is like, excuse me, what did you just say? But they're basically responsible for bacterial conjugation. And what that is, it's almost like a tube that you stick from one cell into another one, and they can transfer genetic information from one to the other. So like that's how some bacteria can kind of like evolve and stuff like that. Eukaryotes, I mean, I feel like we've covered it quite a bit, but eukaryotes don't just include animal cells, they can be protozoa, fungi, plants, all that. There are also some eukaryotic, like you would think mostly like bacteria cause disease, but some eukaryotic cells can also cause disease, like fungi, like ringworm, and parasites. You will learn that more later on. And here's a bit of a table about prokaryotes versus eukaryotes. A lot of you probably have learned this already. And here's another table for just general understanding. Cell cycle. And some spicy memes. All right. Okay, cell cycle overview. We have basically 4 stages. G1, S, G2, and mitosis. Interphase is basically G1, S, and G2, and mitosis is its own thing. Interphase, so it's basically the longest stage, like when a cell isn't dividing, when the cell isn't actively dividing, it just sits in G1 basically, I think. Yeah, it's basically just like slowly growing is what it is. It's just like the cell is duplicating everything, everything's just like growing chunky so that, it can be basically split in half. Yeah, so then you increase the level of transcription factors to enable S phase to continue and get rid of stuff that's prohibiting S phase. Yeah, look, that's basically it. If cells are non-divisive though, like you can see in the corner there's G0, that's for cells that just don't divide. Like nerve cells, once they're kind of created, they don't divide. So they go into G0 stage, which is just a constant state. More interphase. So this is S phase, and that's DNA synthesis. So we will go into more detail about DNA synthesis. Yeah, so basically, the important thing to note is that these are homologous chromosomes. So these are 2 separate chromosomes. These are also chromosomes, but these are sister chromatids. Yeah, so that distinction is important in meiosis. Yeah, so basically DNA is replicated from identical copies, and at this stage, like you're doubling the amount of genetic information, but not doubling the amount of chromosomes, if that makes sense. Yeah. So yeah, in G2, there's just more growth and duplicating everything and to prepare for mitosis. Now, I'm going to go into a bit more detail about DNA replication because I'm not sure if that was last week or this week, but it'll be useful anyway, and some of you would have done it in bio. So yeah, so basically the main things are, like, these are the main enzymes that are used. So helicase is what unzips the DNA. DNA polymerase, yeah, so this is in order. So firstly, helicase unzips the DNA, so like breaks the hydrogen bonds between them. DNA polymerase basically brings nucleotides to the DNA strand and adds them to the DNA strand, or like connects them all together to form a new complementary strand, sorry. DNA ligase, yeah, so it basically, as you can see here, it's not continuous, so you have all these little fragments of DNA. And these have to be bound together by DNA ligase. It's so much. It's not right now. Hay fever is really hitting me hard. Okay, Okazaki fragments. Yes. So this seems to be quite confusing, but to be honest, it's not that bad. The only reason why there's so much text on this page is because I was trying to explain it like a lot in depth, to make sure it makes sense. So I kind of want to go. Can I go back to the previous slide? Oh, well, that's fine. Awesome. So yeah, as you look at it, so as you can see, there's a 3' and a 5' end. So this strand is the 3' to 5', and that strand is 5' to 3'. So the thing is, new DNA is synthesized from a 3' to from a 5' to a 3' direction. So it's made from 5' to 3', and it has to go in that direction because that is the way 3' and 5' kind of works, is that it's like the number of carbons in one nucleotide, basically. So everyone should have done chem. It's like, I have a diagram in another slideshow, but it's like the pentose sugar of the DNA. It's like a pentagon. And the number of carbons, like 3' and 5', is what direction they're facing, basically. So when you number them, like when you were doing organic chem, you would have been like 1', 2', 3', stuff like that. So it's basically, that is what gives the name 3' and 5'. They're not just arbitrary names. Yeah, so as you can see on the leading strand, it's just one continuous long line of DNA can be formed. But on the lagging strand, since it's the opposite direction and DNA must, like, it has to be synthesized from 5' to 3', it has to go in little fragments like that. So basically, like, when the DNA polymerase comes along, it can't go along that way. It has to go this way. But because it kind of wants to be, you know, a bit more efficient, it'd be like one DNA polymerase would land there and start doing it there. Another one would just randomly land there and start doing it from there. This DNA polymerase will eventually catch up to this one. And then it will stop. And then ligase connects to. But in this section, done by that one DNA polymerase, is called an Okazaki fragment, because it's not like one continuous thing. Yeah, so that's kind of what I explained here. Yeah, so it's created from a 5' to 3' direction, but the DNA is read in a 3' to 5' direction. Yeah. So that's what it kind of looks like. An important note is that, you know, it's not going to look like this. Like, it's more likely to look like this. Like, the lagging and leading strand is a bit of a misnomer when you look at it this way, but realistically, it is like still within the DNA helix. So it will kind of look like that. So like if it opened it up there, it'll, yeah, it kind of makes sense if you think about it. Like, when it says origin, it's more like, I guess the origin is where it opens from. So if your origin was here, and you're moving in this direction, that's leading, that's lagging strand. But then when you're moving in that direction, it kind of switches. So if you kind of imagine a line down the middle and like one of these sides is like this, and one of these sides is another this, but you kind of flip them and put them together. That's kind of what it is. Hold up. Before we start mitosis, I'm getting myself a drink. We really didn't think this through. We assigned it by topics, and then like cells is just the entire first week. So yeet. Anyway, mitosis, the thing we've all been waiting for, obviously. I feel like most people already know most of what mitosis is. Like, it's not overly complicated. It's basically, you have a cell, everything duplicates. They pull the stuff to one side of the cell, and then it splits in half. That's pretty much all mitosis is. But of course, they arrange it into stages, and we need to know what those stages are. So, start off with early prophase, which is, yeah. So, this interphase, which is obviously like G1, G2, and S, and then we go into prophase. So prophase, they like to split up into early and late prophase because the nuclear membrane doesn't completely the distinction between early and late prophase, I think it was actually they don't duplicate until, I forgot to put it in, that was stupid. I think it's like the chromosomes don't actually duplicate until late prophase, or they don't. Fuck. My brain is like trying to think back to first year and failing. What's the difference between early and late prophase? Because the chromosomes have already been duplicated. Yeah, I think it is just the nuclear envelope. I think it might just be the nuclear envelope. So like the nuclear envelope isn't fully disappeared until like late prophase. So like everything is still kind of contained until then. Yeah. So it doesn't disappear until late prophase. Sorry. I've been like thinking about second year content for a bit. Yeah, so early prophase versus late prophase. Basically, late prophase, the nuclear envelope has fully disappeared, whereas early prophase, it hasn't yet. You can remember prophase as P equals prepare. Metaphase, it's where everything lines up at the equator. So the equator is basically just the middle of the cell. It's in the middle of the cell, and the spindle fibers kind of attach to the middle bit. So like the bit that connects the sister chromatids is called a centrosome. So it's basically in the middle of the chromosome. And metaphase is also when chromosomes are most condensed. They will like to test you on that. Take note of that. Because everyone will kind of think, oh yeah, it's all condensed by prophase, right? Wrong, it's metaphase. Yeah, and I like to remember metaphase as M equals middle. And by middle, I mean they line up at the middle. Anaphase. I remember in Neutral Bio, my teacher was like, Anna likes to tear things apart. And I was like, well, that's a bit sad for Anna. But, you know, you can remember it like that. Or just A equals apart. I don't know why I didn't just say that, you know. But anyway, my bio teacher was kind of strange. Yeah, so sister chromatids are pulled apart. It's basically entirety of anaphase. And in telophase, they basically just like the cleavage furrow, which is like, if you have one blob, there starts to become a distinction of like two blobs. Yeah, I don't have a picture for it, but that is what it is. It's basically just starting to separate. And cytokinesis is just when they actually separate. Checkpoints. This is important. I also made a slight mistake on the slides earlier because of some conflicting information, but it is fixed now, so it's all good. So there are three main checkpoints. One is at the G1 to S point, so like the crossover from G1 to S. And that checks to make sure that like the cell is big enough to divide, there's enough nutrients to actually sustain like duplicating and growing bigger and stuff like that. Oh no, I didn't change it in this one. We have so many copies of these slides. Instead of S to G2, I will change this later. It should be G2 to mitosis. Yeah, so instead of this, it should be G2 into mitosis. Some people do say that there is one from S to G2, but there are also people that say it's mostly G2 to M. So I'm just going to go with G2 to M. Yeah, so before it goes into mitosis and like fully commits to like duplication, the cell wants to make sure that like all the DNA has been synthesized correctly. You know, the cell is ready to duplicate and stuff like that. Yeah, so that's just to make sure like, wait, do we actually like want to do this basically? And then there is a checkpoint in metaphase just to make sure the spindles do attach to the chromosomes. Mitosis versus meiosis. So we are going into meiosis soon, right after the slide. So yes, the main differences are 2 daughter cells versus 4 daughter cells. So the entire purpose of meiosis is basically to like, you know, it's for this, I've had a mental blank. Sorry, it's for gametes. It's to create gametes. So if you think about it, you know, you can't have a full set of chromosomes from your mother and your father. You would just end up as like not a correct human. Like, I'm sorry, but it just wouldn't work. So obviously you have to cut them in half before they can, you know, become whole again when they combine. Yeah, so you have 4 daughter cells and they all have half the number of chromosomes, and the purpose is to basically produce cells for reproduction. And there are meiosis 1 and meiosis 2. Yep. So meiosis, yes, lovely diagram, but I will start going into it. So I didn't want to like sit here and go through the entirety of meiosis again, because most of it is like the same as mitosis. So I just kind of wanted to highlight the key difference between meiosis and mitosis. Yeah, so this difference is mostly in meiosis 1. So the thing is, right, remember this diagram from earlier? The chromosomes and then, so the homologous chromosomes and then the sister chromatids. The main difference is that we basically have like, in meiosis 1, so by the end of meiosis 1, you already have half the number of chromosomes left, because they've basically separated the homologous chromosomes. Like they've pulled them away from each other, so that you do have at least 1 copy of all the 46 chromosomes. No, you have one chromosome of each homologous chromosome pair in each cell, basically. Yeah, so the main thing is, instead of in anaphase, so this is mitosis, right? So in anaphase, usually these boys will all line up in one line, just like vertically like that. And then the sister chromatids are pulled apart. Whereas in meiosis, the homologous chromosomes line up opposite each other. So like you have them like this, right? Instead of like that. You have them all next to each other, and then the homologous chromosomes are pulled apart. So then in meiosis 2, it happens very similarly to mitosis, in which the sister chromatids are pulled apart. So in meiosis 1, the whole homologous chromosome is pulled apart, whereas in mitosis, it's only the sister chromatids that are pulled apart. So therefore, you are halving it instead of keeping it the same. I hope that made sense, because that's the main point of meiosis, like the main differentiating point of meiosis. Yeah, so then meiosis 2 is exactly the same, because as you can see, so now you have 2 cells, and it happens exactly like mitosis, just with half the number of chromosomes. So yeah, you can see the sister chromatids are pulled apart, and then you have the daughter cells, and there are 4 of them. Yay. That's pretty much it. Okay, quick note on genetic variation. Genetic variation tends to happen like before the chromosomes go into like actually separating and all that. When they line up at the equator, in metaphase, they will do some things to ensure that the daughter cells, like your kids, don't end up looking exactly like you. God damn it, Rachel. Sad times. Phones. Yeah, so basically when they line up at the equator, they're very close to each other, right? So they can just kind of like overlap. So one thing is crossing over. So they're basically just like, their legs will overlap and they'll just switch. Independent assortment is when they line up and like there's no particular order to how they line up. Like the homologous chromosomes will line up, but you don't know like if one of them will be on one side and like say, let's give them arbitrary numbers. Number 1 will line up there, number 2 will line up on that side of the equator, or 2 and 1. So you could have a mixture of like, you don't have a mixture of like all 1s and all 2s. It's like 1, 2, 1, 1, 1, 2, 2, 2, 1, 2 versus like the other side. And random fertilization is just that. A random sperm fertilizes a random egg. Cell cycle regulators. This looks disgusting, but it's something that you do actually need to know. We do have, like, this is kind of a nice way to like clean it up a bit, but there is a way to remember this. It's actually, so this looks disgusting, but basically all you need to remember is DEAB 4221. DEAB 4221. It sounds really dumb, but like if you just say DEAB 4221 over and over and over again, like it'll penetrate. This is how I learned it last year. Like literally exactly how I learned it last year. And then you just remember like, you know, how they line up. So like D reacts with 4 to produce E, and then E reacts with 2 to produce A. So it's like a zigzaggy kind of deal. Kind of just have to look at it, remember it. Cell cycle inhibitors. I hope you learned this already, but basically it is what stops the cell cycle when something goes wrong. A side note is this becomes very important in cancer because when these don't work, it encourages unregulated cell cycle like replication. So then you get like a mass of cells that are kind of fucked and it's cancer. Yeah, so P53 and RB. So P53 is basically a transcription factor. And the main thing is it inhibits the cell cycle. The important thing is when it is on, it prevents transition. So think of it as brakes. When you turn on the brakes, it stops the car. Yeah, so think about these two as brakes. So yeah, so this prevents transition from G1, from G1 to S phase. Yeah, so it's the negative feedback. In approximately 50% of cancers, they like to kind of test some of these stats. So just remember, in 50% of cancers, P53 is mutated. RB is very similar. It kind of has its origins from this type of kind of eye disease. I'll go into it when we cover cancer. Like, yeah. It has to be deactivated by phosphorylation for the cell cycle to continue into S phase. So they're both kind of similar, like this, they're both inhibitory. So when they're activated, they're going to stop it. They're both like brakes. But this one, I can't remember how you turn off. I think you turn off just by like being ready. Like they're just a cascade of signals and they'll turn off. Retinoblastoma is deactivated by phosphorylation for it to continue. Apoptosis. Yes, so you should have had a lecture about cell death and proliferation. So this is the cell death part. So this is just going to cover apoptosis and necrosis. So apoptosis is programmed cell death. It is cell death that the body wants to do. So it's like, it's either from the cell itself. So sometimes if a cell is damaged, we'll just be like, I am no longer useful. I'm just going to yeet myself. So it just yeets itself. And then sometimes if something is removed, so like if an essential substance is removed, it might be like, oh, I need this essential substance. So it just, once again, yeets itself. Yeah, so it is used to destroy damaged cells. And then it can also just maintain appropriate cell numbers to make sure you don't get too many cells. Yes, when you're in the womb, it is used to shape the fetus. Like, I love this picture. I don't know, it just looks so stupid. Like, it's just a circle, and then it's like, wow, it's a hand. And like, so all these things in between will die. Like, all these cells will purposefully die so that you get fingers. And when that doesn't work, you end up with something like this called syndactyly. What time is it? Shit. Okay. Yeah. So apoptosis. These are the steps. They're really weird names, but just kind of mostly remember pignosis. So the first stage is just it shrinks. It basically just like, because it wants to, basically it ends up just disintegrating. So the first one is it really shrinks, like it just condenses. Pignosis is when chromatin kind of condenses really like intensely into like against the nuclear envelope. Karyohexis is when the nuclear envelope basically disappears and the DNA just kind of gets scattered. Blebbing. Now this is the only thing I remembered for the longest time about apoptosis. I was like, blebs. The cell membrane would just kind of all break off into random blebs. As you can think, they just look like blobs. Hence they're called blebs. And then the last one, I like to call it just the phagocytosis phase, because once it's all broken apart, vesicles will just, well, like other cells will come along and just kind of eat up the remains to clean everything up. Definitely write the cell eats itself in the exam. Yes, so now necrosis. Necrosis is kind of, if apoptosis is the good guy, necrosis is the bad guy. So it's unprogrammed cell death. And we don't want necrosis. So yeah, when necrosis happens, the reason why it's so bad is because the cells basically, they don't get neatly eaten up by the cells. It's like when apoptosis occurs, the cell will die and it just gets cleaned up and everything returns back to normal. In necrosis, the cells can swell up and burst, and that just releases the intracellular contents into the environment. And some of these can be quite toxic or harmful when they're outside of the cell. And this can result in like damaging other cells and like inflammation. And then sometimes other cells will like eat up the DNA from that cell, and then it kind of becomes mutated because it got too much DNA, and then it just kind of goes haywire. It's usually caused by external factors like injury, infection, etc. And it kind of looks a bit like if you got really severe frostbite on your fingers, and when they get like gangrene and stuff, and it has to be like chopped off, that is necrosis. Because necrosing cells, they don't function, and since they can't send out chemical signals, the phagocytes can't find them. So you just have a lot of dead tissue and disgusting shit, and often needs to be removed surgically. And this is kind of an outline of necrosis versus apoptosis. Yes, so apoptosis can even be beneficial, like the fingers. Quick questions. Question 1. When activated, P53 and RB control the cell cycle by, do they inhibit stuff or do they enable stuff? Indeed, they inhibit stuff. Thank you. Yeah, I'm getting a phone call. Don't let me. Goodbye. Yes. So, what are you doing? Okay, cool. Yes, so it inhibits progression because when they're active, they inhibit, they act like brakes. I really like the brake analogy, like it just kind of makes sense. Yeah. Question 2, what is not a checkpoint in cell cycle? I really hope I change this. Just sit there and have a quick think about it. Yeah, I'm getting some answers in. Awesome. Yep, so the answer is B. I was meant to change this and I forgot to change it. I changed it on one copy of the slides. I think I forgot to change on the other copy of the slides, but I would change it for later. The correct answer is B. Like there is no checkpoint here. Question 3. What is not a stage of apoptosis? Alright, so the answer is B, syndactyly, because that's just a condition. So that is all from me. If you forgot, my name is Isabel, and if you ever need anything, you can message me or email me or just message the page or anything. Yeah, thanks for that. Now we're gonna have Malika with physiology.
Hey guys, so I'm Malika, and so I heard your physiology lecture didn't quite go as planned, so hopefully this will clear things up a bit. So starting with body fluids, so we're looking at fluid distribution in the body. So approximately 60% is water, and that's split into extracellular fluid and intracellular fluid. So out of the 60%, 1/3 of it is extracellular, and two-thirds of it is intracellular. The extracellular is further split up into interstitial and plasma and transcellular. Although you don't need to worry too much about that, you will be covering it in an ICL at some point if those are up and running. But the most important part is the 1/3 and 2/3. So now we're just going to go through how stuff basically moves in and out of cells, like diffusion and active transport. So diffusion is when molecules move down a concentration gradient. So they move from an area of high concentration to lower concentration. So it increases with a higher temperature, higher difference in concentration, membrane surface area, hydrophobicity, membrane permeability, and low molecular weight. Although not everything is able to cross membranes. So Na and K, so sodium and potassium, are effectively impermeable. This is because while they can pass through the membrane, they're pumped back out again. So it doesn't make a difference. Urea, however, is permeable. So osmosis is passive movement. So it's the movement of water down a concentration gradient through a semi-permeable membrane. So this occurs when so like in the diagram, on one side there aren't any solutes, and on the other side there are solutes that can't cross the membrane. So the concentration on either side is not equal. And so the water moves to the right side to try and balance the concentration. So the movement of the pressure difference is called osmotic pressure. And the last one is facilitated diffusion and active transport. So facilitated diffusion is still down a concentration gradient, so from high concentration to low concentration. It just means that there is like a protein channel or a carrier protein that can help. Active transport is the opposite. It's against the concentration gradient. So it goes from low concentration to high concentration. So electrolytes. So the main thing you need to know about electrolytes is whether they're higher inside the cell or outside the cell. So inside the cell, potassium, magnesium, phosphate, and sulfate are higher. So they're the red ones over here. And outside the cell, sodium, chloride, bicarbonate, and calcium are higher. The main ones to remember are sodium and chloride and calcium outside, and potassium inside. So now we're going to go on to osmolarity, which is the most important part that you need to know, take away from this. So your osmolarity is the total solute concentration in mole per liter. So 1 mole of solute in 1 liter of solvent is equal to 1 osmol per liter, or 1,000 milliosmoles per liter. Generally, we'll use milliosmoles per liter. The osmolarity does not affect how water moves. That's tonicity, but we'll come to that. Another word is osmolality, which is pretty similar, but it's the solute concentration in moles per moles per kg of water. And osmotic pressure, as we talked before, is the pressure applied to the solvent to basically cause the water to move. So red blood cells have an osmolarity of 300 milliosmoles per liter, which is important because you get a lot of questions asking about whether water will go in and out of the cell, red blood cells, when they're placed in different solutions, but we'll go through some questions later. So an effective osmol is solutes that can't move across the membrane, so non-diffusibles. These are the ones that generate osmotic pressure. And like we talked about before, sodium and potassium are effectively impermeable because they're pumped back out, so they can't actually diffuse through the membrane and stay there. And dissociation constant is the extent to which a particular substance is dissociated into ions. So if 75% of AB dissociates, then the constant is 0.75. So tonicity is effective osmolarity, basically. So it is the osmolarity, but only counting the non-diffusibles. So tonicity is what you look at when you figure out whether water moves, where the water is moving, or if the water needs to move. So it's important to note that tonicity and osmolarity are not the same. So something can be isotonic, but not, something can be isoosmolar, but not be isotonic. So red blood cells do have a tonicity of 300 milliosmoles per liter. So here's just a few diagrams to help make it clearer. So on the right side, there's 2 non-diffusible and 3 diffusible particles. So the osmolarity of the solution would be 5 shapes per liter, but the tonicity would only be 2 because the green substance is able to diffuse across the semi-permeable membrane. So in this case, there's 2 non-diffusible substances on the left, and because of that, the tonicity on the left is 2 shapes per liter, but on the right, there's nothing, so it's 0. So that means there is a pressure, there is osmotic pressure because of the difference in concentration. So that means the water will move to the left side. So when there's no osmotic pressure, that means there's no water movement and that there's the same tonicity. So now we're just going to look at red blood cells in solutions. And so starting at the top, the red blood cell has 2 particles inside, but there's 3 outside. So we use hypo and hyper to describe it. So the red blood cell in this case is hypotonic because it has those particles inside, and outside is hypertonic. So that means the water will move out of the cell to go to the area of higher concentration. In the second diagram, there's 2 particles inside and 2 outside, so they're isotonic, so nothing happens. In the last diagram, there's 2 particles inside and 1 outside, so the red blood cell is hypertonic and the solution is hypotonic. So that means the water will move inside to the area of higher concentration. So when we're talking about red blood cells, if a red blood cell is put in a hypertonic solution, the water will go out of the cell, and so we call that crenation, and the cell shrivels up. The opposite is hemolysis, which is when a cell is put in a hypotonic solution and water goes into the cell, so it bursts. That pretty much says everything that I've said, so you can read that later. So here's a quick question. What is the solution? Is it hypertonic, hypotonic, or isotonic in each of the diagrams? So I'll give you a few seconds to think about that. So the solution, the picture on the left is the solution is hypotonic because the water is going into the cell. And in the middle, it is isotonic because nothing's happening. And on the right, it is hypertonic because the water is going out of the cell. So the water will always move towards the hypertonic solution, or whether the cell is hypertonic. So here's a few examples of common solutions. So if you infuse pure water into someone, the osmolarity of pure water is 0. So it is hypertonic in comparison to red blood cells, and the tonicity is also hypo. And because there aren't any solutes, both of them are hypo, and the result would basically be lysis of the red blood cells. For 1.8% urea, the osmolarity is iso because 1.8% urea has 300 milliosmoles per liter, which is the same as red blood cells. But the tonicity is hypo because the because urea is permeable, so it will diffuse across the membrane and go into the cell. We'll go through a couple of questions with this later on, which would probably make it easier to explain. So the result in that would also be lysis. For 0.9% saline, it's isoosmolar because it's 300 milliosmoles, and it's also isotonic because, as we said before, it's effectively impermeable. So it's isotonic. So what infusing 0.9% saline does is it increases the volume of fluid without lysing or crenate or any crenation happening. 5% dextrose is a bit more complicated than the others. Initially, it is isoosmolar, so 300 milliosmoles per liter, but dextrose is used for respiration, and so the amount of it decreases over time. So it eventually becomes hypotonic. So overall, it does increase total body water because it is initially isoosmolar, and it's used for when patients are dehydrated, although it does change its tonicity. So when you get a question of how a red blood cell acts when it's put in a solution, this is the sort of table I would suggest drawing up. So go through, so red blood cell and pure water. So start by writing down the initial osmolarities. So ICF is just the red blood cell, so always 300, and pure water has nothing in it, so 0. And then see if any of the solutes in either of them are diffusible. So nothing will ever move out of the red blood cell. So you're just looking at the solution at that point, if anything is going to move into the red blood cell. And in the case of pure water, nothing's going to move in. So nothing happens at that stage. And then double check the osmolarities. And so red blood cell is still 300 and the solution is now 0 still. And so then you look, if they're equal, then it's isotonic and there's no water movement. But in this case, they're not equal. So the water moves into the one with a higher osmolarity. So here water will move into the cell. So now we'll look at normal saline. So the initial osmolarities are 300 for both. And the diffusibles, there aren't any diffusibles because, as we said, the saline is effectively impermeable. And so the final osmolarities are both 300, so water doesn't move. So saline is isoosmolar and isotonic. If we look at isoosmolar urea solution, once again, the initial osmolarities are both 300, but urea can diffuse into the cell. So that means there's some movement occurring. So we're imagining one tiny red blood cell inside like a bath of water. So when the urea moves into the red blood cell, the osmolarity of the solution effectively doesn't change because there's so much of the solution. So that means inside the red blood cell, you have the 300 milliosmoles you originally had, plus another 300 milliosmoles of urea. And outside the solution, outside the red blood cell in the solution, you have 300 milliosmoles. So that means your ICF currently has 600 and your solution 300. So water moves into the cell towards the hypertonic solution. So urea is isoosmolar, but it's hypotonic. So these are just some calculations showing how to find the osmolarity of all those solutions. They all end up at 300 milliosmoles apart from water, which is 0. So you can look through those in your own time, but you don't need to actually remember how to do them as long as you remember they're all 300. So clinical applications. So we've been talking about 0.9% NaCl, normal saline. So that's given to patients who've lost blood. So it stays in the extracellular fluid when you infuse it, and you can give it to dehydrated patients, but generally you treat dehydration by giving 5% dextrose because it increases the total body water and not just the ECF. In terms of water, never give water via IV because your patients will probably die because the cells will become hypotonic, and as the water will go into them and they would explode. So bad idea. The last one, 1.8% urea. We don't tend to use it clinically, but it's the same, essentially the same as infusing water because of the calculations we did before. So the water will go into the cell and they would explode, and that's bad. So saline and dextrose are used clinically and water and urea aren't. So quick sample question. Take a few seconds to answer this, or a minute. Okay, so a patient is infused with 9% saline, and so the answer is A, because normal saline is 0.9%, and so 9% is obviously a lot more. So the solution is hypertonic, and so the water will go out of the red blood cells into the solution, so the cells will shrivel. And that's it for osmolarity. So I hope that helped. And if you've got any questions, feel free to message me or ask on the chat. Or
Okay, thank you so much to everyone for coming today. Sorry it was a bit slow, we're just still getting used to the Zoom and everything. If you guys have questions, message like anyone or post it in our group chat, group page, and we will get back to you. But hopefully you've learned something. We'll be putting this up online again if you want to review anything. And have a good day, guys, and stay safe.